Hi, this is Dr. Ben Morrill. Welcome to episode 106 of Reptile Genetics Weekly. Glad to have you here with us. We get to have a pretty fun conversation today. I'm excited. Been looking forward to talking about this. We have an awesome guest, a genomics person. So I'm more on the genetics. I think about DNA in small pieces. I don't have a lot of professional experience in genomics, and we have Dr. Warren Booth joining us today that has very, very good genomics background and is currently professor and working in genomics. So very cool to have him be part of this conversation. We've got to talk about dire wolves. We've got to talk about Jurassic Park and, what all are scientists really going to be able to bring back. And so definitely looking forward to talking about that with Dr. Booth and with Kayla and have you all chime in the comments what you all think, what we have to say, and compared to what you're hearing from other sources. We look forward to hearing that. Fun conversation, definitely interesting. But as far as sample updates, as far as I can tell right now, we have a good week this week. Should have fast results going out every day, sex determination going out a couple times. As far as panels, they will not be this week for sure. And next week, if it was next week, it would be the end of the week. It probably will be the following week. So probably looking at about 2 weeks out till we'll have results for panels. But next week, we'll be able to give you an update a little bit more specific. But yeah, just kind of about 2 weeks out. That's all I've got for updates. How about you, Kayla? How are you doing?
I'm good. I am. How about them dire wolves?
There's been a lot of talk. It's amazing to see so much conversation about genetics and genomics and, what is a dire wolf and what isn't a dire wolf. It's fun to see.
And I am a paleontologist. Like literally when I was 2 years old, my mom says that I was looking at her and saying, I want to be a paleontologist when I grow up. I am still a dinosaur nerd. I love Pleistocene animals, and I am ready to have my heart broken today. Well, not really, but also, kind of, but also not really.
Allie in Texas, the one that helps a lot with the samples, running samples there. Her first thing she said about the dire wolf when we were talking about, she's like, I'm just worried now that they're going to bring a T-Rex and I'm going to get eaten by one. And so we'll talk about that. Allie, we'll let you know if that's a realistic fear or not.
Yes. And objectively, even if that were possible, that would be an extremely cool way to go, right?
Yeah, that's nice.
There are worse ways to go. There are less cool ways to go. But anyway, let's bring on our guest today. Yeah. Hi, Dr. Booth.
Hi, everyone. How are you?
Doing great. And once again, I was just like the last time we recorded, I was so excited to bring you on that I actually forgot about business stuff. So you can join us during our business things real quick. We'll knock out of here. On Thursdays, we do our live streams. We do live test results Q&A with our geneticist, Dr. Morrill. I haven't called you Dr. Morrill in like 3 years, Ben. That was weird. And
we're pretty low key on this show. That's for sure.
Yeah, you can even call Ben by his first name there. It's fine.
I prefer being called by my first name, trust me.
Yeah, okay, fair enough. I'll refer to you as Warren then. But yeah, we got that. We are looking for sheds, especially these particular mutations in ball pythons. So if you or someone works with those, definitely send them in. We've got the address to send them to right here. If you want to send those in or any other species or mutation that we don't currently test for, we would love to have sheds from it. Even if it's not something we're immediately going to start working on, those sheds keep for years. So definitely send them in. You don't have to send the whole thing, just a little patch as long as it's dry, because if it gets wet or gets soaked in a water dish for a long time, it's going to denature or destroy that DNA. So make sure it's nice and dry, fresh off the snake, and get us that all the info listed here. So now we are on to our official topic today. So why don't you tell us a little bit about yourself?
Sure. I'm an associate professor at Virginia Tech. My lab really focuses on how organisms adapt and evolve in urban environments, and we study this from a genomic perspective. So we mainly use indoor urban pest insects, and we look at things like the evolution of insecticide resistance and
Yeah.
to model kind of potentially new insecticide targets and so on. But we study how populations establish and spread on a global scale and a local scale. But I've also worked a lot on reptiles, mainly as kind of just a fortuitous project that happened about 15 years ago. I was asked to do a paternity test on a snake, and that snake was a boa constrictor, and it turned out that the offspring were not produced by any of the males of the samples that they sent in, but it was parthenogenesis, and that kind of paper went worldwide. And as a result of that, it kind of just set this ball in motion that every few weeks I would get emails or phone calls from people or letters that they had a snake in their collection and had never been with a male and it produced offspring. And from that, we ended up publishing a lot of different papers on parthenogenesis from boa constrictors to king cobras, and then 2 years ago and into American crocodiles. And we kind of stopped doing the testing on parthenogenesis now for a lot of species, unless there's something really unique, a lineage that, you know, it shows some kind of unusual kind of traits. But we're now interested really looking at the mechanisms that drive it. So we're doing kind of using chromosome level genomes to really understand where the genetic variation exists in these genomes. But that's all side hobby research. That's what I do at the weekends and in the evenings.
That's my light work.
My pay the bill science is related to urban evolution.
Very cool.
And I keep a lot of snakes. You know, I've been breeding snakes for 30 something years, so I've got 100 and something of them at home, and only 3 of them are ball pythons, which Ben knows all about. So
what species did you? Oh, sorry, Ben, you go first.
Yeah, we have the androgen from Canova that you just talked about a few weeks ago or months ago, and then a couple of parthenogens that they sent up as well. So we're going to be sequencing their complete genomes to understand kind of the mechanisms underlying those. So they're sitting at home in a rack alongside some kind of Australian pythons and stuff like that.
Oh, that's so exciting.
Yes. Yeah. And if I remember right, at least some of your online stuff is it's Boa Booth, right? Is that like a website, or is that like your?
Yeah, it's just an Instagram. There might be a Facebook, but I use Instagram for posting all of the stuff that I keep and breed and stuff. So
yeah, so Warren has produced some very, very cool rare species, especially boa species.
Oh, that's awesome.
Yes, and
then I've been on the board of directors of USR pretty much since its inception, so about 15, 16 years. Myself and
very cool.
Jeff Ronnie are the longest serving board members. So we have some other link to the hobby.
Yeah, very cool. And that most recent big publication, well, at least as far as in the media, the crocodilian one, I remembered seeing that pop up, and then it just kept popping up, and it really got. I remember, I think one of the outlets tied that to, you know, this is kind of like a living dinosaur, and it does virgin birth, and, you know, kind of linking that to Jurassic Park and life finds a way, and
I think every paper on parthenogenesis links back to Jurassic Park. You know, Jeff Goldblum, his face has probably been used a lot as a result of it. You know,
I believe it.
So was this the one that went the most crazy, or was the boa one bigger? You know, that was a few years back, but which one was the biggest media storm?
So we've had 3 that were really big. The boa one was the first one, and I think it's because parthenogenesis in snakes had been documented a handful of times. You know, it wasn't novel necessarily. There'd been a paper on Burmese pythons in 2004. We later published a paper in 2014 that kind of discredited that a bit, sadly. But the first parthenogenesis and snake papers came out in 1997 with my buddy Gordon Shewitt publishing a paper that related to garter snakes and rattlesnakes. But this was the first one that kind of really went into it in a bit more detail, using at the time more in-depth molecular tools. And showing that it wasn't just a one-off event. Females could do it multiple times.
Cool.
Shortly after that, I think the following year, we published the first evidence of parthenogenesis happening in the wild. So in this case, what we mean by parthenogenesis is the it's called facultative parthenogenesis, where a female or a species normally produces sexually, but at some point they might switch and produce parthenogenetically.
Mm-hmm.
And that's really what we've seen over the years. Is that's interesting because sometimes part of the clutch or part of the litter could be sexual and part could be parthenogenetic. But it had never been documented in the wild, and it was considered just kind of like one of these artifacts of captivity.
Yeah.
Well, we showed it in 2 different species of pit viper, copperheads and cottonmouths, in that paper came out in 2012. And that was kind of neat because then it went all over the news. David Attenborough talked about it at one of his shows, which is kind of cool.
Ooh.
I was sitting, on the sofa with my kids and then watching a program on virgin birth and then seeing my own work come up was kind of cool. I didn't know.
That is awesome.
And over the years, we published on a handful of different species, moving away from kind of stamp collecting, which is what I kind of call it now. Just another species does it. We know they all do it, right? I'm much more interested in kind of the mechanisms of what drives it. So we published a cool paper looking at the kind of the effects on the genomic architecture in king cobras, parthenogens. We looked at what it does to venom in copperheads.
Mm-hmm.
But the crocodile one was just we took it on because one is a friend of ours, Kessel Dwyer, who people might know that he owns Reptilandia Lagoon in Texas. He owned Park Reptilandia in Costa Rica. And that's where the female crocodile is still. He had called us up and said, look, this female has been isolated since a baby. We went into her one day and she was guarding a nest. We dug up the nest and we found, I think it was 6. I need to look at that paper again. I think it was something like 16 eggs, and I think 7 or 8 looked viable.
Wow.
And whenever crocodile eggs are viable, they have this central band whenever you candle them, right?
Mm-hmm.
So instead of like
picture
in that image, that top right. So just like if we candled a python egg, where you see like the embryo forming, like the red spot. In this case, they form this circular band. And of those, I think those 8, started to deteriorate as they were going through incubation. But one of them got to term and then went over it, never hatched, but they opened up the egg and there was a dead, fully formed American crocodile inside it.
Wow.
And I think we sexed it. I think we sexed it as female. I need to go back to that paper. But we published it. We submitted it to a really high tier journal, and they said it wasn't of sufficient general public interest. But
they were wrong.
Nature and Science did that with a paper early on as well. And we submitted it to a paper called Biology Letters, which is a journal that I really like. For people that want to get into science, this is a journal that will publish like 25 to 3,500 word articles on a whole variety of topics. They're the one that I pick up over my tea in the morning, and I can read about something that's maybe outside my field, because you never know when inspiration is going to come from different areas.
Right.
But they published it, and it's part of the Royal Society, has got a really good press release system with it. Within minutes of it becoming available, I was getting bombarded by the BBC and NBC, every media outlet you can think of around the world.
Yeah.
And it couldn't have happened at a worse time, because that's exactly when I was moving from Oklahoma to Virginia. So I was a professor in Oklahoma for 11 years, and I'd taken on this new position here. And it came out the exact day that I was helping load up a removal truck at my home in Tulsa. And so I remember I was in one of the interviews, I thought it was online, and I was wearing like a sweaty t-shirt, and I was a mess, and I didn't see the BBC. And later on that day, my sister called me and said, "Oh, that interview was great. We watched it and we could see you were sitting on your porch." And I was like, "I was sitting on my porch? I thought I was just doing
audio."
So I was this sweaty mess talking to the BBC about parthenogenesis and crocodiles. A day later, I then drove 14 and a half hours to Blacksburg. So I was down the road from Ben, and every half hour I had different interviews scheduled for that thing. So it just went through the roof, and it continued for about a year. For that there, and when it was ranked as the Royal Society's most publicized paper of the year out of all of their journals, which is kind of amazing.
That is awesome.
And it's fun because it's not my main research, it's just hobby research, right? I funded that myself. It didn't come from grants or anything. That's my own money that did that. All of my parthenogenesis work has always been my own money. But it's with the amount of media, it gets it into the public eye, and it's of general interest. It's a charismatic species. You know, it's likely happening all around us, right? You know, parthenogenesis is not rare. People with ball pythons know it's popping up all over the place.
Oh yeah.
I get contacted with it happening in boas all the time, and it's
Mhm.
interesting because it actually allows people to. Well, what's interesting about it is that I've always said that people that are breeding snakes have a really good understanding of Mendelian genetics, and they might never have taken a genetics course past, you know, high school.
High school, yeah.
And yet they can turn around and work out these 5 or 6 gene combinations in their head and what they should be getting. And I know grad students that are taking genetics courses that couldn't do that.
Yeah.
So yeah, it's just a great way of promoting science, you know, and it's fun and it just kind of shows us that we don't necessarily know everything that's going on in life. Some weird kooky things are happening, and because we don't have you know, wild populations of pattern morph and color morph crocodiles and snakes out there, you know, we don't see these unusual aberrancies that we can in captivity.
Yeah, it's just like you know, just like Dr. We work with them all the time, Dr. Heather
Rafi.
Yeah. And you know, the other people we've worked with for, you know, developing our mutation tests, like they've talked about in their own papers about how the ball python and reptile community at large, keeping these captive species, like, is such an untapped like resource of like, people have been doing like this boots on the ground breeding work for decades. That, you know, I think the scientific community is just now like kind of looking at like, oh, actually there's some cool stuff going on here. That,
Absolutely.
You know, do a science about this, you know.
Yeah, we've in my lab here, we've got boxes and boxes of shed skins from a whole variety of different species. You know, we've got a whole probably most of the Sanzinius populations in the US. We've got probably most of the Boleyn's python samples in the US. And we're going to be making lineages from these here. We're going to be creating lineages to understand genetic diversity in captivity, and then creating essentially a stud book type thing so that people can turn around. And hopefully, whenever people start breeding them more successfully, they'll be able to swap lineages to maximize genetic diversity.
Yeah.
But it's all pet trade derived, you know, and we do stuff. We do work with zoos. We've done stuff with Zoo Atlanta on their Guatemalan beaded lizard projects to work out parentage and lineage for their kind of breeding work. We're doing some stuff for Oklahoma Zoo now based on those as well. So yeah, but the pet trade, the hobby undoubtedly has provided the most material for what we do. We would have none of the parthenogenesis work if it wasn't for that, for me at least, if it wasn't for that one phone call early on from Jeff Ronnie from Jeff Ronnie, but about a boa breeder called Sharon Moore, and she just needed a paternity test. And it was, what's funny about it, it was at a time when academic jobs were really few and far between. They're normally very few and far between. But at this point in time, there was just not a lot of jobs going, and I thought, well, I'm not going to end up as a professor, but I'll probably end up doing paternity tests. And a day later, I get a phone call to do a paternity test. And I thought, that's it, my life's over.
There
it turned out it was just a very fortuitous event that some interesting stuff came from it.
Yeah.
Awesome.
And what's funny, this is the last thing I'll say, is that
you're good. Now
it's really common that ball python breeders and boa breeders all know about parthenogenesis, and they know what's expected from it. So I was, Tommy Crutchfield sent me a link yesterday about a parthenogenesis story on some Facebook page. And it was interesting to see these people responding about how this was incorrect or correct. But a whole load of them got it exactly what it should be expected, you know, based on sex chromosomes and based on, and I thought these are general public that they kind of know what they're talking about. And I thought that was awesome.
It's amazing.
Yeah.
So a man who knows his stuff and has been working with this. I love that. So with that being said, how about them direwolves? What do so Ben, do you want to talk about like just some background if, you know, someone has just now emerged from their rock to hear about this, because it's been all over the media.
Yeah, definitely everywhere, and I would say that probably the most eye-opening thing for me, of course, I hear, you know, here, there, and everywhere, people sending me links and asking questions, and you know, then I was watching an interview with the CEO for Colossal, and he said that this company was started with no scientists at all. But they've brought scientists on, so they have scientists now. But that was very eye-opening to me, because it definitely seems like they have done an extremely good job of marketing and getting their information spread very quickly. It's been amazing how much this has been in the media and all kinds of different things, and headlines and podcasts and just everything in between. So the people they have, like Tom Brady and what's the guy, the movie person, I think it's the guy that did the Lord of the Rings. Anyway, they have these very high, Peter Jackson. Yeah, they have these very high level people that have invested, and it's definitely very interesting. A much more marketable way than if it was started by a group of scientists. That's for sure.
Yeah. And so the thing that's gotten them into the media lately has been the claim that they have brought back dire wolves or something kind of like that. So the three adorable little pups that you see on screen are Romulus, Remus, and Khaleesi. The main two that have been in the media are the two boys, Romulus and Remus. And so so from what I understand, they basically identified genomes of the extinct dire wolves, and found, as it says on screen here, 20 differences in 14 different genes, accounting for some distinguishing characteristics physically of dire wolves. So size, coat, head size, teeth, bigger builds, and apparently vocalizations as well. And so they used CRISPR DNA editing, which we have talked about, I think a few times since we started the show, but especially in our second episode, talking about how some scientists were able to use CRISPR editing to create piebald anoles. And so they used similar techniques to create something something akin to dire wolves here. And of course, media storm, there's of course this Time magazine cover that was the first one that I kept seeing over and over again, saying, okay, no longer extinct, here's a dire wolf. And of course, the NBC 7 San Diego screen grab I got right before here. And it's like this ad nauseum, along with a lot of things think pieces isn't a nice way to put think pieces, and also scientists chiming in okay, you see dire wolves and also quote unquote dire wolves. So what's happening here? Because a paleo nerd like myself is oh my god, that's so cool. I love Pleistocene mammals, and other such nerdy things.
Yeah, so one of the interesting things they're talking about, going to the closest related species that's still on the earth and making those edits that you were talking about, and then having a dire wolf. And so I'll use your doctor name here, because we're asking your professional opinion, Dr. Warren Booth, if there was 20 edits to 20 edits to 12 genes in a complete genome that's 2.4 billion base pairs. You feel like they've brought back dire wolves?
Well, I think it's clear they haven't. I think it's clear that they never will bring back an actual dire wolf. I think what's cool about it, and a lot of people are kind of getting really worked up about it, it's not a dire wolf. And I think Colossal kind of shot themselves in the foot a little bit by saying that the de-extinction of the dire wolf. Well, it's not, but it's on the track for being able to do some really interesting work down the line. 10 years ago, we would never thought this was possible. So the fact that they can edit multiple genes is pretty amazing. We do gene editing work here, and we'll knock out or we'll edit one gene. So doing multiple genes at a single time is pretty fascinating, but also knowing what genes to edit is also really interesting. You know, a paper came out in 2021 that looked at the kind of direwolf lineage, and I think they sequenced DNA from bone of, I think it was around 50 different extinct direwolf, and they were able to show from lots of different areas.
Mm-hmm.
So they were able to identify genes that were variable among them compared to other closely related species and those that were not. Mm-hmm. So they already knew if a gene is already very similar to that, what's happening in an Andean fox or a wolf. The Andean fox is the closest relative, but a wolf is very close to that as well. Then they'll know maybe that's not a gene that needs to be edited. However, if they can go, and because of the canine genome is so well annotated, so we know what the genes are, we kind of know a lot of what they do. So they can turn around and say, right, we're looking for genes that are specifically related to coat color, to cold tolerance, to teeth size, to, you know, muscle structure and so on, vocalization. And they can go and specifically look at the variation within those, you know, 50 that were sequenced early on and compare it to the wolf. And then they know where to make the edits. I think it's a pretty fascinating and pretty amazing process. You know, like one of the scientists that's involved in Colossal is called Beth Shapiro, an incredible de-extinction biologist and geneticist. I saw Beth give a talk probably 20 years ago at a genetics meeting in England, and it was fascinating. At that point, she was talking about extracting DNA from museum specimens, from eggshells and so on.
Yeah.
And it was really fascinating work. We do that in my lab. We sequence genomes that are about 120 years old, right, of insects and compare it to present day stuff.
Yeah.
But to be able to take things that are over 10,000 years old, I think that's phenomenal. And one of the other things that makes it really fascinating is that we think about DNA, right? DNA, your average chromosome might be, 50 to 250 million base pairs long, right? A's, T's, C's, and G's, 50 million of them or more.
Yeah.
Whenever we die, whenever an organism dies, that DNA starts breaking down due to enzymatic reactions. And it gets shorter and shorter and shorter really quickly. The specimens that we work with are like 120 years old. The average DNA length that we get is about 200 base pairs long. So you can imagine trying to get 200 base pair fragments and putting them all together in the right order, span a genome to be able to then target specific genes. You know, so what they do is they use a scaffold. They'll use a genome of a closely related species. And because sequencing technology is advancing at such a rapid rate, we can sequence those short fragments. We can eliminate all of that bacterial DNA that's going to be in that as well.
Yeah.
Right. And then we can start using that scaffold to piece those small little sections like a jigsaw with a template below it of where things go. And then they can focus in and say, right, here's the genes in the canine genome that we know are associated with coat color. Where are the variants occurring? And where can we then make an edit that will then produce a protein that will result in a comparable change? That's amazing. And the fact that they did it with not just one gene, but they did it with 20 edits and 14 genes, I think that's really cool.
Yeah. Oh, and just as an aside, you mentioned something about eliminating bacterial genetic information. So I've heard of this before, but for people who haven't, can you give like a quick explanation of like how do we have bacteria DNA in?
Well, it's purely a result of how the tissue was stored. Like even museum specimens that we get.
Yeah,
they're going to have fungal DNA. They're going to have bacterial DNA. We extract DNA from the air for some of our projects, right? So we can extract DNA from the air to determine what species are present. We're using it not only for that, but we're now using it to determine what mutations are present within specific target genes. Wow. So the fact that you've got a museum specimen that could be sitting in a relatively sterile place, and it'll still have bacteria build up on it, still have fungal build up on it. For what we work on, things like bed bugs and cockroaches, a bed bug, for example, will still have DNA from whatever it was feeding on last in it. So we have to eliminate all of that before we can then take the DNA that's left over, which is the bed bug, for example, in our case, and align it to our genome. So whenever you got a specimen that was not stored in a sterile condition, it was a bone that was buried and so on. Over years, there's been fungal and bacterial growth on it that have penetrated into the bone. That's all going to be problematic. I love this image here, like in Jurassic Park, where they extracted from insects in amber. Beth Shapiro showed that can't be done. A number of people have shown that can't be done. They've They've taken amber, they've smashed it up, they've removed the insect parts, they've tried to extract it, and it doesn't work. Sadly, because I love that initial image of the drill going in and they stick the little needle in, and
yes,
that would be awesome.
right.
Here's the other thing. I think the oldest specimen they've extracted DNA from was a woolly mammoth. It was about 1 million years old.
What?
And I think the DNA fragments at that were about 50 something base pairs in length. Dinosaurs went extinct 65 million years ago. So if it took, in 1 million years, we went from, a very large genome to 50 base pair fragments, it probably tells you that in the other 64 million years since then, we're getting nothing from dinosaurs.
Yeah.
The wish is that they would find soft tissue, that was somewhat preserved, maybe in like permafrost or whatever, but the likelihood of that is just incredible. That they haven't found a living cell of an extinct woolly mammoth yet, so they can't clone a mammoth. And in fact, Beth Shapiro in her book How to Clone a Mammoth, which is really good, you should read it. It's a fun kind of pop science book. She says, I think at the very beginning, she's like, just to set this, make this clear, we're not going to be able to clone a mammoth, but you can edit a genome, which is what's happened here with the direwolf. They've edited the genome to bring back traits that are direwolf associated. That's the plan with mammoths, right? The plan is to engineer, I think, African elephant DNA to bring back traits such as hair length. You've seen it in the woolly mice a few weeks ago. Lipid metabolism for cold tolerance, and they can then be released into areas of Siberia where they will help modify the landscape that has been destroyed due to human-associated degradation and so on. It's all about ecosystem engineering. So the direwolf, I think, was used basically as a model. Can we do it? It's cool you can do stuff in mice, but let's do it something a bit more fancy. And then they take it to the next step.
Yeah. And just as a side note, when we're talking about like DNA degradation, Ben and I talk about this a lot. When we say if you're sending in your ball python to find out your ball python shed to find out if it carries the piebald gene or a clown gene mutation, I should say. Don't let that shed sit in water because otherwise it denatures. Imagine a Pleistocene animal dying of whatever, whether it falls into the La Brea tar pits or is just dies in the middle of a field and is exposed to the elements. Like these aren't things like DNA is fairly fragile. And
fragile, yeah.
Yeah.
Like even with UV light, every day we go outside.
That's
Our
right.
DNA
Yeah.
is getting really badly damaged. That's why we should put on sunscreen.
It's being cooked just by the sun. Yeah.
Right. So that's why your shed skins, I know that people love to have bioactive enclosures and so on.
Yeah.
But UV lights destroy DNA. Humidity. The worst place to store DNA is somewhere that's warm and wet, which is in most, reptile enclosures. Right? You want it cold and dry, and that's the best preservation for DNA. So
exactly.
I've worked on many shed skins. From single genes to whole genomes, and they can be a mess whenever they're just slightly wet. And I get them sent to me all the time, covered in bedding and feces,
growing green mold on it.
Yeah,
bag.
You open up that bag and the aroma that comes out. It's like
it's like Odin Erodia.
That's right.
So. Yeah. So the fact that they were able to extract, I think they said from like some tooth pulp in a direwolf canine, and I think an ear bone was the other one where they were able to extract some DNA.
And it actually works really well. So
yeah,
I finished my PhD 20 years ago, and one chapter in my thesis was looking at I worked on small mammal population genetics back in Ireland. And we were comparing populations and how they changed across a 25-year period.
Yeah.
But these mammals go through these cycles of abundance where you got really high population density and then really low population density, and they cycle every year like that.
Yeah.
And every 4 years they crash really badly due to seed crops and so on. So we wanted to know, what do those kind of cycles do to populations that are spanning different time periods?
Yeah.
And thankfully, my PhD co-advisor's wife had collected samples of the same species from the same area that I was sampling 25 years earlier. So we went, and there was some tissue on the bones that were remnant. but we couldn't get any good DNA from them. But I popped out an incisor from each of them, and I ground that up, and I got really good quality DNA from the dentine of the teeth. So dentine in teeth and bone tend to yield pretty good DNA. I had a student back in Oklahoma, and we extracted DNA from 16,000-year-old American martens.
Wow.
And we were doing some population genetic work, reconstructing genes, and it worked really well.
That's
highly
amazing.
fragmented, so you had to sequence things in a lot different. You had to sequence much smaller fragments. This was using PCR at the time, but we were able to reconstruct mitochondrial genes at the time to look at kind of changes in population structure. So dentine is a pretty good tissue for DNA preservation.
Makes sense.
Kind of it's encased, right? And it's not exposed to the external conditions as badly.
Yeah. And I mean, basically, isn't like the production of plaque and stuff on isn't it basically your teeth are kind of like fossilizing while you're still alive, like at least the production of plaque, I think. I've heard it described that way anyway.
Yeah, I don't know. That sounds kind of gross, but I don't know.
Right? Yeah. But either way, the dentine's very protective, and it kind of makes sense. And like, I guess like the inside of bones, not in the marrow, but like if you have a thick section of bone deep in there, is probably where you're going to find
right
DNA that is less degraded. Or
one of the problems with bone is if you look at it, bone looks very tough and impermeable, but you look under porous. Look at it's really porous. So lots of things can get into it once the flesh is gone. It can get invaded really quickly by bacteria and fungi and stuff like that do bad things to DNA. Dentine, I don't think, the teeth don't seem to have that same issue, especially if they're still located in the skull.
Actually, I think I think a great example of that is like even animals who like rodents and other calcium hungry animals are like gnawing on bones. I think there was actually some other ancient human lineage where they found some teeth in a cave and all the other bones were chewed up. It showed signs that I think a kind of skunk, I believe, was chewing on all of the bones, but it didn't like the teeth. Left.
That's pretty fascinating. Yeah.
Right? Yeah.
They're pretty solid. So
but returning to the direwolf topic, so people have been referring to these as basically just gene-edited gray wolves, which is pretty accurate. So why do you think they chose gray wolves as opposed to, we pulled up this cladogram
box?
Yeah, as opposed to it was I guess
we have a wolf genome, right? We have a chromosome level wolf genome. I don't think we have a chromosome level Andean fox genome.
Oh, well, I guess that would make sense.
That's probably the first thing. But chromosome level genomes aren't hard to do anymore. And I would imagine that colossal bioscience is not short of a few dollars that they could, like we did a chromosome level bedbug genome for about $18,000, so they're not expensive. And that was going through a company. You can do it yourself for about, I think we're working with a group where we're doing the boa constrictor genome and the carpet python genome, and I think they're about 3,000 each, kind of thing. $3,500 each, 4,000. Not expensive anymore. So I'm sure they could have probably worked on the Andean fox. It's probably also what's available in captivity to be able to manipulate. There's lots of places working with gray wolves. There's probably not as many places working with Andean foxes. Even though Andean foxes they're not a species of concern. They're not a conservation concern issue. But there's just not a lot of places working with them that. I don't know. It would be interesting I'll have to find out, because at a meeting last year I was talking to a few people that came from a friend's lab that all work for Colossal now. The next time I see them, the next meeting, I'll have to ask them why they chose the gray fox and not the gray wolf, and not the Andean fox.
Yeah.
But it's probably just ease, but it's probably just they're easier to get.
It would make sense.
Level genome.
And granted, like we touched on the topic of de-extinction for ecological engineering, like with the woolly mammoth. And from what I've read, both from Colossal and also like other people, grappling with the ethical bird's nest that is ecological engineering and also de-extinction. From what I understand, they're not making quote unquote dire wolves to release them back to the wild. Like Romulus and Remus and Khaleesi will never know the wild. And I think it's just kind of a proof of concept, possibly just for the market. I think maybe part of the reason they also chose gray wolves, maybe because it was a marketing idea. They talk about how, they actually make a huge part of their marketing the fact that we are in a anthropogenic extinction, like mass extinction right now, caused by humans, and how they make a big part of their marketing. Here, look at all these partners. Save the Elephants, Conservation Nation, all of these advisory members who are concerned specifically about species who are still alive. And so, people are asking, okay, why are we trying to bring back why are we bringing back extinct Pleistocene mammals who do not have a niche anymore? Dire wolves were hunting, woolly rhinos and these massive megafauna in the North American ecosystem that existed when they were alive, that evolved with them, as opposed to, animals that are currently at risk of becoming endangered or extinct because of humans. And it seems like an odd way to go about it. Like, why are we going this way instead of just trying to take care of the animals we have right now?
Well, for me, they're two different paths, right?
Yep.
Species are on the verge of extinction or going extinct due to human actions every day or very frequently, right? That's totally different than what scientists can do and are doing, right? Because we can engineer mutations into genes doesn't mean that that's going to save that species.
Right.
I've reviewed a lot of papers recently on population genetics of species in Madagascar. And those, various gecko species are going extinct because populations are fragmenting and becoming smaller and smaller until they start making habitats go bigger and bigger again. It doesn't matter how many genes you engineer, the species are still going to go extinct.
Yeah.
With something like the dire wolf, right? So something like, with the idea of resurrecting a mammoth-like creature, that can actually play a role, right? One that was hunted to extinction by humans, they believe, right? Based on the evidence to date, it looks like pretty much humans resulted in the loss of that species.
Yeah.
There's landscapes that can be remodified from the release of elephants that are modified for that. So they actually have an ecological engineering role. They can actually help reduce carbon impact and so on.
Yeah.
So these species can actually play a major environmental role. So I see them as two totally different paths, and I totally understand when people are saying we should be investing our money into saving species. I totally agree with that. But sadly, that's not happening. And a reason for that is a lot of governmental action. Whenever you lose, major areas of habitat for oil drilling, for whatever, for urban living, for it's not you're destroying habitats, and therefore the species that were there can now no longer live there. So we're reducing resources, reducing habitat size, reducing the number of animals that can live there within the new area, and therefore ultimately you're going to lead to these extinction vortexes due to inbreeding that will occur as a result of small populations. So that's something that needs to be addressed on a totally different level.
Oh yeah. And I think that ties very much into the words marketing keep coming back to this, and you're completely right. And I think I forget which article it was I was reading in my research for this, I guess. But they were talking about how basically like standard or for conservation efforts, traditional conservation efforts for extant species, a big part of that is getting the funding for it. Funding for like pushing for pushing or lobbying for governmental regulations or actions that protect habitat or, help more. And also, of course, just, like the money and effort it takes for captive breeding programs or what have you. Traditionally, it hasn't been enough because humans like to put their money toward things they're excited about. And unfortunately, not everybody's excited about protecting, I don't know, the Eastern hellbender, which is, just a big old like snot otter up here in Appalachia, like a lot of people are way more excited about the thought of like a dire wolf or a woolly mammoth. Or if you're a billionaire, and you're like, what cool thing do I want to put my name on and my money for? The thought of being the man behind funding the bringing back a woolly mammoth is way cooler than, oh yeah, I put some money toward this gross looking salamander thing. And I hate that. For one, on an emotional level, because I love hellbenders, but that's kind of how we work.
And like a lot of the money that's being, I would imagine that they're generating, a lot of the funds that they're generating, they might be targeting woolly mammoth or whatever, but that money is also being used for these other projects that are allowing us to see it's advancing it's advancing science dramatically.
Yes.
Developing methodologies for editing genes, because that ultimately can be used for humans, right? For editing human embryos where, there's major health conditions that are associated with single mutations and so on. That's a phenomenal thing.
Yeah.
It's got application potentially for conservation down the line. What happens if we are getting to a population that's become so low due to inbreeding that there's a number of key genes that are modified that are resulting in stunted growth and so on? They can now target potentially those genes, knock out those mutations, induce a mutation that switches that deleterious mutation off. That's really beneficial. So there's actually long-term conservation potential for these projects.
Yeah, the American chestnut is
right.
Huge,
for the rest of science, we're screwed because funding is becoming just horrendously bad to get.
Yeah.
We're in a time where they're looking at collapsing scientific funding.
Yep.
And yet scientific funding for the projects, they advance science dramatically. They have impacts on human life. So the fact that they can generate money for these kind of charismatic species, for the charismatic projects, I think it's fantastic because it has top-down effects. We will see the long-term effect of that in a very short period of time. The beneficial rules in humans and in species of conservation concern.
Exactly. And that's why I'm both like on one hand, the media storm about, dire wolves are back. On one hand, it's easy to kind of like poo-poo the like it in general, but at the same time, the scientific and ecological implications of something like this are so exciting. I think we mentioned the American chestnut and bringing back, helping a keystone species of North America that was decimated by blight. That could be a huge thing. North American species have evolved alongside the American chestnut. That is a niche that could be refilled. I know that cheetahs and red wolves are a great example of genetic bottlenecks. And the stuff that they're doing up at Colossal could be used to help these species, in addition to captive breeding projects. So like there's like even it, I kind of joked at the beginning. I was like, I'm ready to have my heart broken, but like there's I can see how this is with Colossal. I can see how this is kind of a marketing thing to springboard off much more, what's the word I'm looking for? Much better is
more impactful.
More impactful, yes, more impactful projects.
Yeah. And hey, that's what we all do. If we're trying to get funding for something, you got to use the thing that's really charismatic and really exciting and really sexy and
exactly
and people like that. It's like for the work that I do on indoor urban pest insects, the National Science Foundation doesn't fund me for that work, but they fund me because I can use them as a model system to study mitochondrial evolution or recombination rate or use of museum specimens to understand temporal change.
Mhm.
They don't give a stuff about a bed bug. So if I went to them for money for that specific, they're going to say it's species specific, and there's no point. So the fact that the collector can go along and say they can have those couple of big charismatic species, the dodo, which itself will be an amazing thing because
Right.
modifying something that actually is in a hard-shelled egg is really problematic. It hasn't been done to date, to my knowledge.
Yeah.
So if they can bypass, and I've heard rumors that they have, if they can get through that step, then that has major conservation issues for California condors, and a lot of these species that have really been impacted by humans, like California, a lot of the due to DDT, for example, in the 1940s and 50s.
Yes.
So I'm excited by what they're doing. They're advancing scientific method and scientific discoveries at a rate that would be impossible in the regular research lab, I think. Just because of the charismatic species, the fact that people will donate for that there. And you get these foundations, you will get rich people that want to, we've seen Jurassic Park and the idea behind that there, right? The rich millionaire that wants to bring back a park, it's kind of cool. But realistically, there are people like that. My position, my research position here is funded from a family who were in pest control, and they donated $1 million to fund my lab.
Oh, that's wonderful.
And it's because of their interest, so you get these people these philanthropists that are interested in advancing science, and they're able to, I'm all supportive of that. Getting money is very, very difficult for scientific research.
Yeah.
So whenever we can get something like Colossal and the backing that they're getting, not just from private individuals, but from foundations, the fact that they've got foundations that are associated with wolves and elephants and so on, they're obviously clearly seeing the potential of this for conservation down the line.
Definitely.
Yeah, I haven't gone onto their website in a while. I need to actually go. My fear is that I'll get sucked into this tangent, and I'll lose about 3 days just going through their website.
Right. This is my first time going through going through their website as well. It is interesting and they're hitting the marketing really, really well with this. This is fun to look at. And I just from a science communication standpoint, I do have some mad respect for that, because they are trying to and again, this has to do with both funding, but also just getting people excited about science and biology and ecology again is also real. I mean, that's super important. There are incredible things going on in universities and research centers worldwide, but a lot of people, without science communication, people don't quite understand, and if they don't know about it, they can't care about it or love it.
Yeah, and that's why I would recommend. So I don't know Beth Shapiro, so I'm promoting her books, but she doesn't know anything about me. But the How to Clone a Mammoth is excellent. And last year she released another book, I think it was last year, the year before that, called Life as We Made It. And it really brings these kind of advancements in science into the kind of your sitting room. Years ago, I bought my brother a book. My brother's also a PhD population geneticist, and he's in Scotland. And I bought him a book years ago, and it was How to Clone Your Hamster. So sorry, How to Fossilize Your Hamster, another experiment for the armchair scientist. But it brought kind of real life science. So for example, it brought real life science into kind of that story that is funny, and I'm considering developing a course here at the university behind the science of Star Wars.
Oh my gosh, yes.
It's not about Star Wars. It's about how we can take something that people are into and then to really understand science behind it. So for example, Han Solo and carbonite, and relating that to hibernation and brumation, and what happens metabolically and what happens, during this process. But it's a nice kind of way to say, and this is what that relates to, right? But it's trying to bring science to the layperson, or to the person that's not a scientist, or as the armchair scientist, which I think a lot of people on Facebook are, thankfully with better genetics understanding. But I think having something like Colossal and the way they are able to promote that research brings it to the layperson, which I think is exciting.
Yes.
Yeah, they're doing a great job of that. That's for sure.
Yes. And I think it's so interesting. I'm still on the dodo page, and it shows off it shows off the exciting, cool, like the exciting, cool, and sexy stuff about bringing back the dodo, this iconic extinct species, and then the very next thing is here are the endangered species, like on this island, and species they want to save that are related to dodos, because dodos are basically, okay, I'm saying this is broad strokes, but they're basically big old pigeons.
Yeah, that's what they are. Yeah, their closest relative is a pigeon.
Yeah, which I love. I love pigeons so much. So I think that is extremely cool. I like that they're doomed as a dodo. That is.
Well, that is true, but look at the passenger pigeon.
Yeah.
The passenger pigeon in North America was a species that was so abundant that there's reports of them of the skies being blackened for days as these flocks were flying over from west to east, right? And then it just turns out people thought they were delicious, and they hunted them to extinction. The last one died in a zoo in the 1950s, I believe.
I think so, yeah.
But they were hunted to extinction from species that had existed in the billions, basically. Flock sizes in the millions, and within a very short period of time. And it was related to the advent of guns and also the advent of the railroads, because they could actually stay ahead of the flocks and then just start shooting them down as they went along. That they resulted in the extinction of a species.
Yeah.
The problem with that is that if you de-extinct a passenger pigeon, how are you going to get the numbers up to where they start flocking and they have the actual behaviors of passenger pigeons? That's one of the fears with any of these de-extinction projects, right?
Yep.
You de-extinct a mammoth, but a mammoth would have fed on the feces of the mother to build up its gut bacteria and so on, right? For part of it. And they would have learned behaviors from the mothers that would likely have been different from elephants. So, that's why another reason you're not bringing back the mammoth, you're bringing back a mammoth-like, a mammoth-looking, it's faux mammoth, but it's, there's a lot of other issues that are associated with de-extinction that I don't think we will be able to get around. And that's the learned behavior, the nurture part of it.
Exactly. This is just a nerdy thing on my end, but like as a child, I think I remember at least one point I was thinking as a kid, I was like, I'll never know what an actual dinosaur sounded like or how it acted. I got like teary-eyed thinking about it. I've been a nerd all the way down, guys. But yeah, you're completely right. Even if we somehow get the nature part of the nature and nurture thing right, which we never will because we don't have the full genome of these animals. But even if we did, like the nurture part of that, like that is such an important part of an animal's development, and like life and death critical even for a human child's development.
But even if we have a genome, remember that even if we have a genome and we understand what genes do, it doesn't mean that those genes were functioning. Right? So genes are switched on and off through this epigenetic regulation, right?
Yep.
Where the DNA unwinds and it exposes that region and it gets replicated and so on. We don't know, because we've got a genome sequence doesn't mean that we know exactly when these were happening, at what stages of development they were happening.
Yep.
We do this work with insects because insecticide resistance is both genetically controlled, but also epigenetically controlled, where the environment actually triggers resistance. And we're working out methods to actually understand what genes we're doing from 100-year-old dead specimens. We're working on that there.
Yeah.
But that's where we have fragments of DNA where we could say, right, these are 250 base pairs long. We understand whether they were functional or not. You can't do that with a 1,000,000-year-old woolly mammoth. I'm not aware that they can do it with a 15,000-year-old, direwolf. So again, because you got genes, because we know the genes because we know the mutations in the genes that were present, doesn't mean that we know whether they were actually functional and at what stage in their life they were functional. So it creates other issues.
Exactly. This is just a side note, but I think I see a lot of, I'm pretty sure this is AI art we're looking at.
I hope so.
Yeah, like I think it's not the first time I've seen something that looks like AI art on here, which we won't get into that hamster ball of stuff, but I've just seen that a couple of times just as a throwaway
But with the mammoth, they're starting off easy. They're starting with the woolly mice, right?
Yes.
And that's kind of cool because what they've done with those is really neat. They've modified a couple of key genes like fibroblast growth factors, which are associated with hair length, right? So I think these little guys have got 2-inch long hair because they've been able to modify mutations into these fibroblast growth factors.
Yeah.
And then they've also looked at lipid metabolization. So they've done they've modified lipid metabolization genes, so therefore it should relate to cold tolerance. They've modified a melanocortin gene, which will relate to hair color. So these are all steps to get to that mammoth-like creature. The thing is that the reproductive rate of a mouse is, you're going from embryo to adult in about 28 days.
Yeah.
It's a bit more difficult with an African elephant. You're on about 2 years, right? So of gestation alone. But if they can start modifying these genes in mice, then it kind of sets the scene for what can be done with elephants down the line.
Yeah.
And of course, we know that elephants are also species of conservation concern. So what the techniques they're learning here is not just all about, wasting an embryo of an African elephant to turn it into something different. In time that those same techniques will be used to help modify deleterious mutations out of African elephant genomes to help preserve species lineage longevity.
Yeah.
Which is so cool.
Now, is it going to happen in snakes soon? Probably not too often. Probably not too soon. We're not going to be able to turn around and create our 11 gene ball python using CRISPR and this technique. I think that's beyond us at this moment in time.
Yeah.
But give it 5 or 10 years. The group in Georgia did the albino anoles. Now from what I know, they all died. So, so they didn't survive very long.
Yeah.
I think there was a pied anole or something done as well.
Yeah, we did an episode on that.
Right, but so that's pretty costly and pretty complex.
Yeah.
But we never know where we're going to be. Like I 8 or 9 years ago, I didn't think we'd be at this stage for shed testing for snakes. But with how rapidly technology advances, sequencing technology, it's changed that dramatically.
Yeah, it's a different ball game. I remember being in biochem class, and I think this was 2000. He was talking about the Human Genome Project and how long that was going to take, and all of a sudden, like 2, 3, 4 years later, it was completely different ball game.
Yeah, like the original human genome was 10 years and $10 billion, right? And it just created contegs. It created fragments, didn't create chromosomes.
Yeah.
Really, they were complete, and now we're doing chromosome level genomes in a really short period of time. But I'm currently writing a paper from my PhD. So I did this work 22 years ago, right? This actual chapter, and I should have published it years ago. And we just got swamped with other things. So it uses 7 microsatellite markers, which are highly variable markers, and they're informative, but I tie it with dental morphology and all this stuff. Got microbiome stuff, so it's really cool for the story it tells. But we just had a paper accepted on kind of urban pest evolution, where we use 7.5 million markers, not 7. So it's 7.5 million variable sites versus 7 variable sites. The story, it changed, and that's just in 20 years. So and we're in that exponential curve where technology is advancing at such a rate. To be able to use museum specimens the way, like we literally just a handful of weeks ago, we were extracting DNA from museum specimens where we had the insect and we gave them the insect back at the end of it, and yet we'd extracted DNA from inside it. We digitized it. So we got a high resolution image of all aspects of it, and then gave them it back at the end so they can put that back on display, and we've got its DNA that we then sequenced to chromosome scale.
That's amazing.
Freaking insane. And it costs, I think it's about $120 to do that there.
What? That's amazing. That's really cool.
The world is changing so fast. So
yes. And just because the question will come up, and I guess Ben had somebody ask about this too. Like
Allie, this is for you, Allie.
Yes.
No T-Rexes.
No T-Rexes. No T-Rexes.
So it goes back to what I said earlier. And God, I'd love to go to a park and see this kind of stuff. I've flown over the landscapes in Hawaii, where these were filmed, and I've flown
Right.
in areas of Costa Rica that look just like this here. I mentioned that DNA from the 1 million year old mammoth was really, really fragmented. I think it was 50. It might have been even shorter than that there, base pairs in length. And that's only 1 million years old. It's 65 million years. The last dinosaur went extinct at 65 million years ago or thereabouts. So that DNA is in such short fragments that you might get 3 or 4 base pairs in a row, but the likelihood of that being duplicated somewhere else in the genome is really high. You're not reconstructing those genomes, sadly. But they are doing work to modify chickens to change beak structure and to change kind of how they walk and so on. But
turning teeth back on,
right? And that's kind of cool that it can be done. That you can turn on and off genes.
Yeah.
But I don't think we're going to be reconstructing a T-Rex anytime soon. Of course, right now probably in a lab somewhere there's a T-Rex embryo forming, but I don't think it's happening.
If someone gets eaten by it, maybe it'll be me, and that'll be
worth it. Totally.
Exactly.
I'll eat my words then, but I can't see us having a dinosaur anytime soon. Sadly, I think it'd be kind of cool, but sadly not.
Yeah.
And I don't think we're going to see a mammoth really anytime. I think we're going to be 5 or 6 years out before we see a hairy elephant.
Yeah.
So, but it would be cool to see that as well.
It would be. So there's a lot of things. So there's as far as the media hype, it is a bit of a bummer, but there's still so much excitement that still makes my heart go pitter patter looking at this stuff. So it's cool stuff.
Big step.
Yeah. And as I say, science is making big steps because of technology. And that's really exciting. We just need to maintain funding for science to allow those big steps to keep happening.
Yes.
We don't want to go on the decline. We want to keep going up.
Yes. Exactly.
Fund science, folks.
Yes, fund your local scientists. We like them. And just one last note: you mentioned how the albino anoles died pretty quickly, which is very sad. But that was from changing one mutation, that being OCA2 gene.
Yeah, but remember what happens with a lot of these here, right? So with using things like CRISPR, they're creating a guide sequence of DNA. They're using molecular scissors to cut it, but there is also the possibility that guide is also replicated somewhere else in the genome, because unless they have a chromosome level genome, which there's not a lot of chromosome level genomes out there, they could be missing fragments where that target is also, and it could be in an important gene that is switched on later on in development that could then lead to death. So what with chromosome level genomes, it certainly makes the development of these guide RNAs better. And we'll probably see fewer of those detrimental effects, which is probably why the dire wolf has been relatively successful.
Yeah, and they're making it to, I guess, sub-adult stage is where they're at now.
They're 18 months old now, I believe. I think they were they like October of 2020? Were they?
Something like that. I think so. 23, maybe.
23 was it? Yeah, I'm not sure. I thought they were up to a decent age at the moment.
Yeah, which is extremely impressive because it made 20 different edits.
Right.
But knowing that apparently, like you said, whole multiplex genome, larger genome edits makes
so they be making genome. So within a single lineage, they're not just making a single edit; they're making 20 edits. I'm assuming that there's not whether they made embryos prior to this that had 3 and then added 2 more and then added a few more to it. I'm not sure.
Right. Yeah.
But, it suggests that they made multi-gene edits in a single lineage, in a single generation.
So
hopefully, I'm sure we're going to see a paper come out about it.
Oh yeah, I'm excited for that. Mostly because I can have you guys help translate what I'm reading, because I will want to read it. But
and since Peter Jackson's a part of this, we might see a blockbuster come out about repopulating ecosystems with gene edited animals. I could definitely see that coming.
Yeah, but you are seeing stuff. You are seeing this stuff change the way some movies are being made, right? So if you think about the first Jurassic Park, they
Oh, yeah.
talked about, the fact that they use frog or amphibian DNA, and that's why the females were able to change sex. And at the time, obviously I wasn't working on parthenogenesis, but later on when I started working on parthenogenesis and what we know about sex chromosomes,
Yeah.
that could also be parthenogenesis caused those males to be produced.
Yeah.
And then I think in one of the more recent ones, they talked about using varanid DNA to reconstruct, and they talk about parthenogenesis, I think, in it, right? They mentioned the potential for it. So
that's awesome.
I thought it's kind of cool how that is changing kind of popular culture as well.
Yeah.
So yeah, I think it's fun. It's changing the way we think about things. So
awesome. Yeah. All right. Well, that was I think we covered some really, really good ground today. Let us know if, I know this is a bit of a departure from our normal topic, but I mean, we have our friendly genome scientists and geneticists here. Of course, we got to talk about it. So let us know what you think. If and when the Colossals paper comes out, if you want us to talk about that and, get Warren and Ben back in on talk about what they see and what their thoughts are, let us know in the comments. And of course, comment, subscribe. That way the algorithm knows that we're awesome and we can do some science education, science communication of our own. Warren, where can people find you if they want to find more of your stuff?
Scientific papers on my lab website, which is booth-lab.org. And then my Instagram is just Boa Booth.
Boa Booth. Awesome. Yeah. Okay. Now this time for real. See you guys later.
Bye.